Welcome everyone to the program of hands-on fNIRs for Motor and Auditory Research. My name is Mariana Ceci and I will be your host for this lecture. This event was brought to you by the Santos Dumont Institute, the University of São Paulo, the Federal University of Rio Grande do Norte, the State University of Campinas, the Federal University of ABC, Brain Support, and NIRx Medical Technologies. We would like to thank all these institutions and the professors who took part in organizing this event. Here, you will listen to the lecture of Mr. Rickson Mesquita, who is a professor at the Physics Institute of the State University of Campinas, located in São Paulo, Brazil. Mr. Mesquita has a degree in physics obtained from the State University of Campinas and obtained a PhD in science from the same institution. He has worked as a visiting researcher at the Massachusetts General Hospital and as an associate researcher at the University of Pennsylvania. His current areas of expertise include biomedical optics, light transport in diffusive media, optical properties of tissues, functional spectroscopy, and photodynamic therapy. Let us all welcome Mr. Mesquita.
Hey, thank you, Mariana, for the kind introduction. Thank you, everyone, for attending. Good morning or good afternoon, depending on where you are. So I want to start by thanking the organization of the event. I think this is a very important step towards kind of advancing NIRs and making NIRs more popular among scientists, mainly not only in Brazil, but across the whole world. So in this spirit, I guess what I'm trying to do here for the next few minutes will be to explain a little bit of our recent efforts about the fNIRS signal and mainly try to go through some steps towards reproducibility, right? So that's a question we keep asking ourselves for a long time. We all do science, and science should be reproducible, right? So it's not science if different people across different places in the world do the same thing and they get completely different results. So, but there are reasons why this may happen, and that's what we've been trying to investigate and trying to overcome over the past few years, I guess. So, okay, so that's my goal, and I'll try to just give you a brief introduction, a brief motivation, and also a little bit of the guidelines that something that we learned over the past 10, 15 years on working with NIRs in our lab. So I guess I don't need to say much about this, right? But I guess it's always good to remember that there are different spatial scales you can investigate the brain, right? Going all the way from the molecular level to the system level. And in the very microscopic scales, you do have a little bit of what you get is mostly structural information and basic functional information, how the neurons work or how the cells communicate to each other, things like that. And on the kind of more macroscopic level, you're more interested in questions like how the brain works and how is the functional organization of the brain. Why does the brain consume so much energy? So it's more related to the brain function and brain physiology rather than just structural information, right? So there's a lot to be covered when we talk about brain function, but I guess the most and you can use optics to cover all those scales across completely different techniques, right? But I guess when we talk about NIRs, we're mostly talking about the macroscopic level and trying to answer questions about how do the different regions of the brain work together to perform a function, and questions related to the metabolism and oxygenation across different regions of the brain. So when we talk about NIRs, we are mostly kind of the bottom line here of this slide. Right? So, and there are a couple of different ways and a couple of different protocols you can try to use to probe the brain. I guess the most widely used and performed protocol to investigate the brain, at least with neuroimaging, at least at the macro scale, right, is performing what we call a functional neuroimaging protocol, which essentially is you perform, you are interested in understand how the brain functions under a certain task, a certain stimulation. It may be an external stimulation. Most of the times it is an external stimulation, but it can also be kind of an internal change or an internal task, an internal stimulus that can trigger some brain functions, right? So, and because, and I'll talk about a little bit further about this, but because the neuroimaging techniques are actually very low in the signal to noise ratio, right? So you are actually trying to image the brain and find a signal in the middle of a lot of noise. What we try to do is to repeat the same task under the same conditions and compare kind of whatever we are seeing with any neuroimaging technique with the baseline when the person is not performing the task, right? So that's basically kind of the essential experimental protocol and the simplest experimental protocol that one can envision when talking about neuroimaging, right? So you have whatever neuroimaging technique, and by the way, there are a lot of them, right? I guess the most widely used and the most widely known is the fMRI technique. I guess the oldest one is the EEG, electroencephalography. And NIRs is just one among several different neuroimaging techniques that we can use to probe the brain. But whatever the technique we're using, what we are always doing is actually collecting the data at different regions of the brain over time. And as we actually go along with this part, with this experimental protocol, you try to, at some point, compare the task with the baseline and try to see differences, and mostly regions that would be correlated with the task and or activated with the task. That's typically we call it activation maps, right? So just to give you an idea of how this works, right? So this is a very kind of illustrative video that was performed with fNIRS. So we do have a lot of sources and detectors on the head, and these sources and detectors provide any signal, which is here just qualitative, and it just fluctuates a little bit, mostly because of noise, but you don't see anything changing. But from the moment that the subject starts performing a motor task, what you do see is that some regions of the brain change, right? In this case, there is a brain activation, and this correlated with the stimulation in the left hemisphere, close to the motor cortex. From the moment it stops, it also goes back to the baseline, and things should kind of go back to a steady state during the baseline. And if you start another task, you see a different region of the brain that can actually change with the stimulus, right? So this is kind of the most, it's the simplest thing that you can do with fNIRS and neuroimaging, right? And the reason you do this and you correlate kind of these changes with the tasks and with brain function is because we know that changes that we measured with NIRS are sensitive to the changes in neuron activity, right? So basically, whatever you're measuring with fNIRS is the hemodynamic part. It's basically NIRS is sensitive to the absorption, to the hemoglobin absorption in tissue, right? And since we have different types of hemoglobin, we are mostly sensitive to the oxyhemoglobin and deoxyhemoglobin absorption in tissue. So you're always measuring at the end part of the spectrum, which is kind of the hemodynamic response. And we know that whenever you have a neuron activity, right, which you can always measure with electrophysiology parameters, that will demand an increase in oxygen consumption. And this oxygen is carried by the hemoglobin, and specifically oxyhemoglobin, around to the tissue that you need, right? And that's something that we measured kind of over 10 years ago, I guess 13 years now, which is a very simple experiment that really shows that whatever you're measuring with the optical imaging here is related to what neurons are doing at the electrophysiological level, right? So this is just a very simple experiment, which we actually performed a whisker barrel stimulation in rats. So you have the whiskers of the rats, right? And every whisker is controlled by a very specific region in the mouse brain. And what we do is actually every time you actually deflate or move the whisker, you see a change in the electrophysiological signal, and correlated with this electrophysiological signal, you see an increase in oxyhemoglobin and a decrease in the oxyhemoglobin. And if you sum the total hemoglobin, you have the green line, which is a decrease, which is also an increase in total hemoglobin, which should be proportional to blood flow, right? And you always have an increase in blood flow that sustains this increase in oxygenation, right? So we know these things are linked, and therefore, every time we see an increase in oxyhemoglobin and a decrease in the oxyhemoglobin correlated with a stimulus, there is a great chance that this is related with neural function, right? Which is typically what we want to measure in neuroimaging, right? And NIRs is very useful when we like to use NIRs because of a lot of several characteristics or features that the technique has, right? The first one is safety, right? NIRS does not use any ionizing radiation, does not use any exogenous contrast. Everything that you're measuring is just hemoglobin, which is inside your blood, right? So that allows one to measure continuously 24 hours, 7 days a week if you want to, because there's no safety concerns related to the technique, right? Second, because we're always talking about lasers and detectors, light detectors, which thanks to advances in microelectronics, right, can be very small and very portable. We can design near systems that are very small and very portable and also very wearable, right? So that allows you to perform or think of experimental protocols that can be outside the lab even, right? So this is one very interesting and I guess one of the very first examples from NIRx, where they actually measure the brain during biking, out completely outside the lab, and the whole system was inside the backpack, right? And you were measuring brain function with a probe that is underneath the coat here. So that allows one to think of a lot of completely different experiments in completely different fields that had nothing to do with neuroscience at all. For example, the human sciences, right? Education, I guess you'll hear about this after my talk, and a lot of completely different things and fields that we had never had the opportunity to really address questions from the neuroscience perspective. Right? Another thing is that because they are portable, they are very flexible, right? And you can think of systems that will be very insensitive to motion compared to the other neuroimaging techniques we have, right? We will see that NIRS is also sensitive to motion. Motion can actually be a big problem in using fNIRS as well, but it's a bigger of a problem when you're talking about fMRI or EEG or MEG, magnetoencephalography, and things like that. So when you think about other techniques that you have available to probe the brain, I guess fNIRS is one of the least sensitive to motion, although we still need to be careful about it. And that allows you to assess populations that are not easily assessed with other techniques like infants, and probably you heard a lot about this yesterday. Right? And the last thing, right, that I can highlight is that NIRS is relatively inexpensive, right? Mainly compared to other techniques, it's a kind of a very affordable technique that you can have to investigate the brain. So all these features make kind of NIRS really nice and really powerful, and at the same time, really wishful, right? We really want to use this to measure brain function, right? From the moment you have an instrument, you can collect data acquisition, and this has been there's been a lot of progress on data acquisition, mainly from the companies that are selling these instruments, right? They are really making a lot of effort and a lot of advances and making these techniques really plug and play for us users to kind of just put the probe on and start acquiring data, acquiring NIRs data, right? However, from the moment you have the system and you collect the data, there is a long journey, it's a long road until you actually get NIRs results that you can really interpret them, right? And I guess that's kind of the hardest part, because most of commercial instruments we are very used to, mainly if you are in the medical field, right, or the biomedical field, we are used to just get systems, press buttons, see the results, and we don't even question what the results, what's kind of how the results came from. We just assume that whatever it's spitting out, it's typically the truth, and then we can interpret the results, right? I guess what I plan to show here is that NIRs is not like that, right? There are kind of a lot of steps on the way, and there are a lot of things and a lot of details that you really need to be careful about before you actually start interpreting whatever results you get. So just some of the steps with some of the kind of solutions we came up over the years, right? We as a field, not we, our lab. Okay? So in order to find brain activation after you collect fNIRS data, I guess the very first thing you need to think about the optical probe, right? So you have some sources, some detectors, and this will allow you to cover one region of the brain, or hopefully more than one region of the brain, but will certainly not cover the whole brain. And even if it covers the whole brain, you have a bad spatial coverage, right? Because we are just starting to think about systems with a very high number of source and detector called high density systems. But most standard systems that we can acquire today, they are not high density. So you need to really think about how you want to use and how you want to kind of arrange your sources and detectors to make sure you can have a good coverage of the regions that you really want to study, right? And over time, the field has come up with a lot of different tools that we can use to help us design optical probes, right? I guess Atlas VNIRS is probably the most used ones. It's also the hardest one in this sense. But we do have a lot of good tools that came up over the years, right? So Array Designer is a very nice tool that you just say how many sources and detectors and the regions you want to monitor, and they just try to optimize the source detector location for you. There's Fold, which was created here in the Federal University of ABC by Guilherme Zinho, which is a really nice tool as well, that once you decide where you want to put your source and detectors, it tells you how sensitive you are to the different regions once you have a fixed one. And more recently, I guess from later last year, there is a recent development from Fold called DevFold, which allows you to do the same thing that Fold does, but in neonates and kind of very infants, which have a completely different brain anatomy, right, in terms of size. So this is the very first thing that you need to do. And fortunately, already, there are some good tools that we can use as long as you know what's the research question you want to address, what are the constraints, right? And basically the number of sources and detectors you have to work with, you can think of a smart way to design and arrange your probes, right? After that, you need to collect the data, right, with a NIRS system. And I can't tell you how important it is to understand and to assess data quality before you start the measurements, because collecting fNIRS can be really tricky and it can take some time, right? So typical measurements can easily take around 30, 40 minutes just to set up if you want to really have good data quality, right? And you don't want to go through all this work and in the end find out that the data you collected is mostly garbage. Right? So there are tools also that you can assess data quality. I guess Phoebe, which is kind of a very clever tool from the University of Houston, Luca Polonini's lab, which basically make sure that whatever data or whatever signal your system is detecting, it is coming from the brain, and it has a good signal compared to the noise that it's intrinsic to the data, right? So there are a couple of different metrics, SNR, the signal-to-noise ratio you can easily calculate. There is also most instruments use the coefficient of variation, which is okay, but it's not ideal, right? But then once you collect the data, then it starts the whole processing data, right? And there's a lot of things here that you can do. You can try to deal with motion artifacts. You can try to inspect your data for motion. I'll try to kind of show you that there's a lot that comes with the signal that it's not related to what you're looking for, to the brain response. So you have to do some effort to actually separate this. And finally, you need to really come up with a solution or with a framework that will correlate the changes or the signal you're measuring during the task with the signal you're measuring when you're not performing the task, when the subject's not performing the task, right? So you have to find the hemodynamic response that is associated with the experimental protocol you're doing, right? And here you have a lot of steps, and there is a lot of things that you can do. And also, over the years, there are a lot of packages that we can use. Homer is probably the most widely used package for NIRs analysis, but we have a lot of different ones. NIRs Storm is becoming very popular. Analyzer is also becoming very popular. If you are more into Python and not MATLAB, there's NIRs ME and things like that. And it can also write your own libraries if you wanna, if you know how to program or knows someone in your lab that does, right? So, and after that, the last part is you wanna know what you got, right, from the experiment. So you wanna, after finding the hemodynamic response, you wanna show the hemodynamic response. You can do that just by looking at the channels, right, the source detector combinations that you measure. You can reconstruct this onto a real brain, and there are a lot of softwares that can do that too. You can just project your results onto a map like we've done here. So there are a lot of things that you can actually do to look and analyze the data, right? So a lot of steps, but if you really don't understand them, they become stones on your way, right? And it's important to really, you don't need to be an expert in all of this, but it's important to recognize that there are these steps. They do something to your data, and it's better that you understand what they can do to your data and what how powerful these instruments can be to kind of really find a very small signal in a kind of a sea of noise, but also it can also be confusing if you don't understand what you're doing, right? So I guess my plan to the rest of this talk and to the next, I guess, 20 minutes is just to show you some, or just to give you some, I wouldn't say advice, but maybe just share with you whatever we learned over the past 15 years, 17 years, working with fNIRS and trying to work in right, to advance the field and make sure people are getting the results they want, so that they can actually interpret from a physiological perspective, from a medical, clinical perspective, from a biological perspective, whatever your field, from a psychological perspective, whatever your field is, right? So the first thing, and I guess the first advice is you really need to understand the physical principles of the measurement, right? Even if you don't have a kind of math background, an engineering background, or the physics background, you don't need to go through the math and understand the whole math behind it. But there are principles from which the system works and the technique works, and you need to be aware of this, right? So just to give you a very glimpsy idea, I don't have an intention to really bore you with physics, but we all know, and I guess the main idea of physics comes from what we have been doing since the 19th century, right? Which is related to modeling the transport in different media. And I guess the simplest one that we do, and it's very common in chemistry, right? If you have a homogeneous media and you shine light, light will be attenuated. And this amount of attenuation is proportional to the, basically, to the molecules that you have in the media that can absorb the light you are shining, right? And you can quantify this by the absorption coefficient, right? So basically, just shine light with a very intensity at the top, measure whatever light you are getting at the bottom of the medium, right? A distance L. And whatever you're getting can be related to this capacity of the molecules in the medium to absorb light, which is, in physics, we call it the absorption coefficient. And since the 19th century, we know that this absorption capacity of the medium is related to the concentration of the molecules that you have in the medium, right? So this is very commonly used in chemistry to understand the composition and the concentration of molecules in any medium, right? However, when you are talking about biological tissue, you have one problem, and this problem is scattering. It's just not, light will not be just attenuated by absorption of the molecules, but it will be completely scattered by the different molecules that you have in the tissue, by the different cells, the different types of tissue you have when you shine light. And what this does is actually whatever we were doing when we just have a medium that absorbs, now it's not going to work anymore because you have to account for the scattering, right? And even the kind of profile of propagation completely changes. We use it to learn that light propagates in a straight line, right? Well, when you have scattering, the microscopic profile of light propagation is not a straight line anymore. It's more kind of a diffusion of light across the tissue. And because it's a diffusion of light, you can still kind of mathematically treat this problem as a kind of a diffuse problem, right? And that's really what you want to do. But the problem is actually, once you shine light in a medium that scatters a lot of light, like the biological tissue, you have to untangle the situation and whatever is related to absorption to whatever is related to scattering, right? So the real problem from the physics perspective is to separate the contribution of absorption from the contribution of scattering to the light that you were measuring, right? And this is not an easy problem, and people have, a lot of smart people have spent a lot of time and even kind of life trying to advance and figure out solutions to this problem, right? So the easiest one is to kind of get what we know from chemistry and kind of the Beer-Lambert law and modify it so that it can be used in a scattering, highly scattering medium like biological tissue, right? And that's basically the foundation of mostly fNIRs studies or fNIRs systems, right? And basically whatever we were getting before, now we have to adjust because of scattering. And this is one very good important thing to think about and to always keep in mind. This is a kind of simplification of the problem, of the real problem that is going on. And you cannot simply separate absorption from scattering, right? So, and this only works if you are in the diffusion limit, which means that you need some kind of spaces and you have some space constraints so that this is still true. If you try to do a measurement, an fNIRS measurement, with sources and detectors very close to each other, depending on the absorption and the scattering of the medium, right, of whatever tissue you're trying to measure, you will not be within the diffusion limit. And therefore, will you get a signal? Yes. Will the instrument measure something? Yes. Will that mean something? The answer is no, because whatever you're using to analyze the data does not work in some limits, right? So you gotta really understand this because this imposes constraints on, for example, the sources and detectors you have, and the space between the source and detectors you have, right? Then this is only useful because since you cannot separate absorption and scattering, right? This is only useful if you try to relate changes in this intensity you're measuring to changes in absorption and scattering. And you can only do that, again, I don't have any intention to explain the math here, but I do want to point out the assumptions we make every time any package does something to treat your data, right? And the other assumption is that it only works if you are thinking about small variations over time, right? If you are thinking about big variations in tissue over time, these will not hold either, right? And that has a lot of implications. For example, if you are trying to measure stroke, ischemic stroke, or a big hemorrhage, right? Then you may have a big change in absorption and a big change in scattering. So that may not be true anymore. Whatever you are doing to the data may not hold anymore, right? Because it may not be a small variation in absorption or a small variation in scattering anymore, right? And the latter part, right? You always assume that scattering does not change very much compared to absorption. So that allows you to kind of relate the intensity you are measuring over time to the changes in absorption and therefore the changes in hemoglobin concentration, right? That is also true. That's only true if you have high reduced scattering compared to absorption. And again, there are situations where that may not be true. Again, if you think about clinical situations, I can think a lot of diseases where the scattering and absorption will vary a lot, and therefore this kind of approach will not give you results that make any sense, right? And to make things worse, yeah, you need to think about how long or what's the length that light travels through the tissue, which is something that you can only guess with continuous wave NIRs, which is the most widely technique, right? Another big thing that it's important to keep in mind is that, well, you shine light to tissue, and light kind of diffuses through tissue. Part of this light comes back, and that's what you measure. Depending on the distance between the source and the detector, you have the kind of the depth information that you are getting is related to the distance between the source and detector, right? So this is a kind of very nice simulation that shows that as you kind of keep the detector far away from the source, the deeper you can go, right? If the source and detector is very close, you don't go very deep. So you don't have any information above few millimeters into the tissue, right? So typically what we try to use is the 3 centimeters because it's enough to have some information around 3 centimeters deep down from the scalp, which is kind of whatever closest to the cortex if you consider all the layers between the scalp and the cortex, right? This problem has been deeply investigated from a physics perspective, right? And since 1995, for example, there is a paper that shows how this kind of the mean depth is related to the source detector separation. And also it's related to the absorption and the scattering of the tissue. So it really depends on the tissue you're measuring, the depth penetration can be different, right? If you're measuring the brain, there is a lot of absorption, so that will not allow to go really deep. If you're measuring, for example, the muscle, the absorption is lower in muscle, so you can actually get a deeper information from deeper tissues than what you would get from the brain, right? I guess, but one thing that it's very important to remember and to always keep in mind is that if you are at 3 centimeters source detector separation, which is the standard for at least adults experiments, right? Less than 6% of the signal you detect comes from the cortex, right? All the 94+% that you are collecting and you are measuring is not related to what you were trying to measure, right? So this is huge, and people really ignore this, right? So and you should never ignore this, right? So over 90% of the signal you're detecting, it's not what you want to measure, right? And you really need to think about this, right? Because whatever you, so if it's not what you want to measure, it's still coming from the brain and from the tissue, right? Whatever you're measuring. So what you need, what it is that you are measuring, and it also has relationships with hemoglobin, right? So the second part is actually you really need to understand the physiology underlying the measurement and not the physiology underlying the protocol. Typically, you design an experiment because you want to understand some physiology or some function at some level, right? So that's the physiology behind the hypothesis of the study, which you should have, of course. But beyond this, you need to understand the physiology underlying the measuring, what the measuring is really measuring, right? And well, if you think about it, right, when you shine light from the scalp and the brain is all the way beyond, so there's a lot of tissues in between. And these tissues also have blood vessels, and they will also have their own hemodynamics. Hemoglobin is also flowing through flow, right, in these tissues in between the scalp and the cortex, right? So even the scalp has its own hemodynamics that you'll be measuring. And most of the signal that you are collecting is coming from this data, right? And if you think about the brain circulation, there's a lot of data and a lot of vessels, a lot of arteries that will fluctuate over time the amount of hemoglobin because, well, they respond to arterial pressure oscillations and things like that, right? And in fact, most of the changes, right, most of the control in blood flow comes from outside the brain, right? So even the fluctuations, if you can separate, yeah, and if you can trash the 94% of the data, you still have a lot of fluctuations that it's not related to the brain, right? So one way that we learned over time to deal with this is, and to try to separate that, right, the 6% of the signal you are interested in with the 94% of the signal that you are not interested in, is to kind of removing and regressing out this information. So one easy way to do this is to have one very short channel, right, very close to one detector, very close to the source, so that this detector does not measure the brain. And if you have another detector that measures both 6% of the brain, 94% of not brain, you can use the information from the short detector to kind of separate, to try to regress right, all the 94% you are measuring, and it's not that's not what you're interested in. And that really works to some extent, right? So this is a very interesting study that we just published, showing that, yes, there is a lot of, right? So if you think about just the data that you have across different regions of the brain, and by regressing out, you remove a lot of these contaminations with this global signal, right? With the systemic physiology that comes outside of the brain. But it does not remove everything, right? And we know that because if you use this regression and then you remove data from the physiology that you can collect independently, you still have some stuff to remove, right? Mainly in oxy and total hemoglobin. The oxyhemoglobin is less sensitive to this, right? Because it's mostly on the venous side, and it's not very responsive to global systemic physiology. Like oxyhemoglobin. And that's another mistake that people do quite often, right? So they typically report just oxyhemoglobin, which is the mostly contaminated signal, right? When we should really be reporting deoxyhemoglobin data, right? Because it's less sensitive to whatever you don't want to measure, right? And the reason people typically mostly report oxyhemoglobin, not deoxyhemoglobin, is they do that when deoxyhemoglobin doesn't show any change. And that could actually raise a yellow flag that whatever you're measuring is not coming from the brain. It's not what you think you are measuring, right? If you don't see any changes in deoxyhemoglobin, that's probably one good reason to suspect that you're not measuring brain function. Whatever you're seeing is coming from the 94% of the signal that's not related to the brain, right? I'll point out that sometimes these systemic changes are correlated with the tasks, and they can misguide the news interpretation as well. So performing a short channel regression like we just mentioned here is a good practice, but it will not solve and will not work for every protocol, right? So every time you have a protocol, for example, in physical exercise, that you bump and you kind of hike, you get systemic physiology all the way up, that will interfere with the measurements as well. So this is one example that we were actually performing a strength exercise experiment. And what we do see is actually, it looks like you have a very nice activation profile. Oxyhemoglobin increases, the oxyhemoglobin decreases. But then when you look at the heart rate, you see that this change, heart rate also spikes all the way up, right? From 80 beats per minute to 120. And it's also correlated with the stimulus. And if you account for this heart rate change, you see that you don't see a brain function change at all, right? So all this change that we initially see is related to the heart rate and all systemic physiology. And once you remove the heart rate and systemic physiology, you don't see the changes that you saw at first, right? So sometimes it doesn't really work, right? But sometimes it's good, right? So you can think of whatever you're trying to trash, like systemic changes, right? These 94% of the data, although it's not coming from the brain, it may be useful for some studies, right? So this is one study that we just finished, right? That relates and shows that these systemic changes that people usually trash, they do contain information to cerebrovascular diseases like carotid stenosis, right? And you can use not the neuros part, because the neural part doesn't have any change, but the systemic change can be useful sometimes, right? And again, even after accounting for extracerebral contributions, the cerebral hemodynamics is modulated by intrinsic and extrinsic factors, right? And this is very important because, and probably this is one thing that I just focus now, is that there's a lot that can change the signal. It's not just the neurovascular coupling, right? You have systemic factors that will change the artery flow and therefore will drive changes in oxygenation, right? And this is very important. This is one very nice study performed in mice 10 years ago, right? That shows how the oxyhemoglobin, or the total hemoglobin, but oxy and deoxyhemoglobin changes to pressure, to changes in pressure, right? So depending on the stimulus amplitude, you can induce changes in blood pressure, and these changes in blood pressure can drive the changes in oxyhemoglobin. And at the same way, small decreases in blood pressure can drive changes in oxyhemoglobin and spike changes in deoxyhemoglobin, right? So, and this is not related to brain at all. This is just blood pressure changes, right? It's not due to neurovascular coupling. It's not related to neurons spiking. It's just the kind of central system, the autonomic system, nervous system, driving changes that you are measuring, right? And the interesting part is actually this tends to be very common when the brain is not completely maturated, which is the case in infant studies. So this same study showed a very nice response of kind of the fNIRs signal, right? When the mice is very young and you don't see an increase in oxygen, decrease in the oxyhemoglobin. So trying to think and translate whatever we do with adults to infant studies will not work because the brain may not be maturated, right? Using GLM approaches and things like that to infant studies will not lead you to somewhere very meaningful because you don't have good a good neurovascular coupling in the developing brain yet, right? And the last thing that I want to think about when you think about the signal is actually really, I know that NIRs measures oxygenation, and that's what we want to do, right? That's what we can do, but it's really what we should be targeting on, right? Because the cellular activity will demand brain metabolism, and brain metabolism tends to decrease oxygenation. But on the other hand, the same cellular activity will increase blood flow, and this increase in blood flow will increase oxygenation, right? So, and whatever you're measuring, when you are measuring oxygenation, which is what you measure with NIRS, it's also what you measure with fMRI, whatever you are measuring, it's a result of a competitive effect between the decrease in oxygenation forced by brain metabolism and the increase in oxygenation forced by blood flow, right? And most of the times, blood flow wins, and that's why you see an increase in oxygenation, an increase in oxyhemoglobin, right? But that may not be the case depending on the task you're doing. And this competition may also be very well matched if you're thinking about cognitive tasks and higher order demand tasks, because they are not, they are more kind of fine changes in the brain, right? And so this is just one example that we measure with in a sheep, spinal cord in a sheep, right? So from the moment they actually have an occlusion, right, in the sheep, you see that the oxygenation is slowly decreases because it's a competitive effect between whatever it's coming in from flow and whatever it's being consumed from the tissue, right? Whatever, if you are trying to measure blood flow, you immediately see the decrease, right? So really, and that's why even in the optics, diffuse optics field and fNIRS field, you see people trying to come up with solutions to measure blood flow. DCS is mostly no NIRS technique that measures flow because it's a cleaner signal. So it will give you, you provide users a better marker to see whatever is going on in the brain and not NIRS, not oxygenation. Right? So another quick thing is that we need to avoid motion whenever possible, right? And sometimes that's not possible. Infant studies, if you don't, if you avoid motion, you don't do any measurements, right? So you need to know how to deal with this when it's inevitable, right? And but fortunately, there are some solutions to this, right? And there are basically two big types of motion artifacts that can be induced. One is kind of very spikes in the data. Another one is kind of this changes in baseline. And we came up with a solution that can actually change and deal with this type of artifacts. And this is just a validation of the solution, but I guess I'm not gonna have time to explain now. But it's that's something that we can do, right? And then I guess most importantly is actually we need to accept that fNIRS has a low signal to noise ratio. And every time you are measuring something with low signal to noise ratio, that will lead to high variability. And high variability has a lot of implications to group results, right? So trying to measure a couple of subjects and looking at group results may not be a good idea if you have a high variable cohort. And that's may be also the case with fNIRS, right? So just to give you an idea of the problem, right? This is a very simple finger tapping test, 2 seconds of finger tapping test. And if you just get very few subjects, like 5 subjects, you can get a very good average response, right? So you see a change in around the motor cortex, in the channel's position around the motor cortex. You see an increase in oxyhemoglobin, a decrease in the oxyhemoglobin, which tells you more than an increase in oxyhemoglobin, as we already mentioned. But if you get 1 subject and try to do the same experiment with the same subject at 3 completely different days, you get 3 completely different results. And even if you try to get the same subject to perform the same task at 3 different times of the day, of the same day, you get 3 different results, right? So, and the reason being, whatever you're measuring is kind of immersed in a sea of noise that responds to systemic physiology. And as we can do a lot to try to eliminate that, but even doing this, right, there is a lot of systemic physiology that governs the signal, right? And there's a lot of variability that you can actually do the signal. You can try to decrease this variability by kind of tracking the optical positioning, and that's we showed this a few years ago, right? By just keeping good track of where your signal is, you can actually get more consistent results across different days or across different times of the day within the same day, right? So, I mean, I know that may be a little bit pessimistic, right, and not very, I don't know, a good something that encourages everyone. But I guess I wound up saying that there's light at the end, right? And like The Little Prince says, right, which is a book that I guess everyone should know. I must endure the presence of few caterpillars if I wish to become acquainted with the butterflies, right? So there's a long journey and some stones in the way before you actually get what you're looking for. And if you actually try to be careful with this, you can actually do very interesting things. So this is one example of measuring plasticity in infants, where you can actually see specialization. And we could only do this, and we could only see this after being careful with the probe design and correct emotional effects, thinking about the non-maturation of the brain, not doing a GLM in this case, and trying other approaches, right? And you can also compare full-term infants with preterm infants and try to characterize the delay in the kind of brain development, in this case, in the sensory motor cortex. So this is something that we can do. This was done with the Federal University in Minas Gerais, Brazil. This is a very nice study that we also saw plasticity following a traumatic brain injury. So you can measure the brain, right, and before and after a rehabilitation procedure, and see the specialization that it's kind of widely known that should happen during rehabilitation. We can actually measure that after taking care and really thinking about the signal and the pre-processing, right? You can even do resting state studies and show that whatever results you're getting, it's very reproducible and comparable with techniques like BOLD, right? And you can find a lot of different resting state networks, right? And this resting state network can be so reliable that it can actually recognize subjects based on the resting state patterns with over 99% accuracy. And that's something that we've been working on now, like trying to recognize subjects based on the kind of fNIRS profile, like the Minority Report movie, if someone shows it, right? So I guess to end, I'll just say that, I mean, NIRs is a very valuable tool, don't get me wrong, and you can really apply it to several scenarios and circumstances. However, right, like kind of the Batman movie, right? Great power, or the Spider-Man, I don't remember. The great power comes with great responsibility, right? So you really, you won't get meaningful results. You can get results, they are just not gonna be meaningful. You can even try to explain your results, but they are not really coming from the brain. So you really need to spend some time to think about the origins of the signal, the physical origins of the signal, the physiological origins of the signal, right? You have to ask basic scientific questions, and you have to find ways or use ways that have already been published in the literature to overcome limitations, right? And in the end, I can tell you, it's all worth it, right? So with that, I'll just finish, and I want to acknowledge my lab, because I was a fortunate guy presenting the data here, but really all this work was done by students and postdocs in my lab, along with a lot of other people from different labs that we collaborate with around the globe, right? Mostly in the US and Canada, but also a lot of different places in Brazil as well, right? So if you want to know more about us, you can just check our website. We try to keep it as updated as possible. And we also have a YouTube channel that we try to publish some educational material about some of the issues that I described here and we try to work on. Okay, so you can also follow us on social media, both Facebook and Instagram, on Lab Unicamp. I will try to talk that, and that's all. I hope you guys. I didn't demotivate anyone, but I just encourage people to be better than what we've been doing. And I'm happy to answer any questions if we still have time.
Thank you so much, Mr. Mesquita. Now we are going to follow up with the questions from the audience. The first one is by Sheila Ballant. She actually has two questions. She says, the first one, congratulations for your lecture, Professor Mesquita. What are the main differences in cerebral hemodynamics in infants and impacts on the protocols and analysis of the fNIRs in infants? The other question is, are there specific and different patterns with the signal-to-noise ratio in infants? Thank you.
Yeah, so starting with the kind of the first question, right? There are, I guess, the whole brain function idea is based on the assumption, right, of the neurovascular coupling. And you have kind of, right, a neuro response to a task, and this neuro response is coupled with a hemodynamic response that we can measure through oxygenation, oxyhemoglobin, deoxyhemoglobin. And most of the times, how this coupling works is something that we don't know, right? And most of the times, what we do is we model this coupling through a hemodynamic response, which is typically a kind of a gamma function that increases in oxygenation and decreases in deoxygenation. It has a peak several seconds after the task onset or something like that. So it's a model, right? And it's a model that works. And we know that we have extensive literature on this in adults. And the problem with measuring infants is actually their brain is developing, and this neurovascular coupling is not completely filled depending on the time, right? So we measure kids starting at 20 days of age, right? And go up to 1 year, right? So most of the times, this neurovascular coupling is not completely formed. And therefore, this model that we use is not going to work, right? And if you're trying to use this model to analyze infant data, really what you're measuring, right, before anything, is the regions of the brain that are fully maturated, which is good. I mean, if that's what you want to measure, okay. But most of the times, that's not the research question that I see researchers have, right? So you got to consider that the brain is not fully maturated in infants, and because they're not fully maturated, they will respond they will be very susceptible to arterial pressure, for example, right, and to drives in systemic physiology. So I guess it's important to be aware of this, because that will, I guess the main thing is actually whatever we do for kids, for adults, it's not going to work for children, right? I guess that's the main thing. And what works then? Well, that's a good question. That's what there's a lot of people trying to think about this, right? Our solution is never assume anything in the brain, in the infant brain, and always try to measure something, right? The reason we assume a model response comes actually from the boat, from the fMRI field, right? So we translated whatever they were using here in fNIRs research, and it works well for adults, but it does not work for infants. Right? So I guess that's the first part of this, and the second part, I guess, great. So there are, I guess, as I mentioned, motion is a big problem for infant studies, right? And we know how to deal with this, fortunately. And I guess we have a pretty good solution for this in the literature, right? So I guess motion is one very specific problem. Most of the times you can keep an adult to stay quiet, but not a kid, not an infant, right? Not and unless it's asleep, and that's most of the times not what you want to do in some experiments. Another thing that it's important to consider is actually, I told you that right, 94% of whatever we're measuring does not come from the brain, right? In infants, the distance between the scalp and the cortex is smaller, and therefore you get more data. It's not 94% of trash, right? Of garbage. It's going to be less. So you are more sensitive to the brain in infants than you are with adults, and that's a good thing, right? Because if you get more of what you want to measure, more signal of what you want to measure, that will make your life easier, right? So I guess those are the two main important things that I can see and I can think on top of my mind now. One helps infant studies, the other it's a concern.
Okay, thank you very much for your answer. The next question was asked by Sergio Novi, and he says, thanks for the excellent talk. What do you think about adding additional measurements to remove motion, such as accelerometers? Since the complexity of the system increases, would it still be worth it? Thank you.
Well, it's I think it's still worth it. It does increase. It does add another layer of complexity, right? We've done that a couple of years ago by kind of plugging accelerometers and in our optical probes, right? And I think it's a good way to make sure what a motion artifact is. One of the big problems in whatever solution you see in motion artifacts is trying to identify the motion artifacts. And although there are automated ways to do this, it's always dependent on three parameters that it's kind of research dependent, right? So one researcher may choose one parameter and that will make the kind of algorithm more sensitive to motion, and the other one may trigger a different parameter which will make it less sensitive to motion and things like that. If you have an independent measure of motion, you can somehow use this to identify or to at least rectify, right, whatever you were getting in terms of motion artifacts. So I think it is a good thing to have it. On the other hand, I understand it's a trade-off, right? Because you're adding more complexity to the data, but it's always what's the price you want to pay. I guess that's the question every researcher should do before starting a protocol, right? So because I mean everything that I said here and all the solutions that we come up with and other groups come up with as well, they depend on having some equipment in addition to the fNIRS measurement, right? You need to collect systemic physiology independent of the fNIRS measurement. You need to collect, you need to track the position of electrodes with a digitizer, which is independent of the fNIRS connection. So it's a trade-off, right? So if you want to really have really good, reliable data and really understand, go deep down what you're measuring, you probably will need all of this. If your question can be answered without so much details, maybe you can kind of get rid of some of this in the auxiliary instruments that you would use.
Thank you very much for the excellent talk, Professor Mesquita. We would like to thank you all that are watching this video, and please don't forget to subscribe to the Santos Dumont Institute's YouTube channel so that you can have access to these and many other contents related to neuroscience, neuroengineering, and scientific research.